make sure we're recording. Well, good afternoon. Thank you for coming. It's our pleasure today to have Dr. Dongkyun D.K. Kang, who's an assistant professor of optical sciences and biomedical engineering, who's come to the University of Arizona from being an assistant professor at Harvard Medical School, did his PhD work in Korea, and is going to tell us today about a topic that's too long to read, so we'll let him read it.
All right.
So, Dr. Kang.
Can everybody hear me? Oh. All right. Let me try to adjust this a little bit. Hello? Hello? Not sure if I'm doing the right thing, but I guess pretty much everyone can hear me. So, all right. Cool. Hi, everyone. So as introduced, my name is Dongkyun Kang, and I typically go by DK, so you can call me DK. That kind of makes my life easier. So it's very nice, meeting you guys. And it's a real pleasure to present my work at my new home. So today we're going to talk about my research topic, which is spectrally encoded confocal microscopy for comprehensive and low-cost in vivo cellular imaging. So there's quite many keywords here, and I'm going to try to cover in one keyword at a time so that towards the end of the talk it kind of makes sense to everyone. So yeah, let me start with the confocal microscope. I'm pretty sure pretty much everyone's familiar with some sort of microscope, but just to reiterate, confocal microscope uses a point source, typically is a laser, and focuses light down into very tight in a small spot inside of the tissue. And then what's coming back from the focal point, either reflected or fluorescence light, comes back and then focused through this pinhole and detected by the detector. So that's pretty straightforward. This is called confocal microscope because there's a conjugate relationship between the focal point and the pinhole location here. And what it does is that the light coming back from the focal point has easy time going through the pinhole and gets detected. And all the other light coming from this particular plane, for instance, is focused somewhere else and then gets rejected by the pinhole. So what it does is that it can generate a very clear image from one very thin focal plane, and that's often called optical sectioning. So in reality, what it does is compared to conventional light microscope, where you don't really see too much of details when the tissue is thick and have a lot of scattering going on above and beyond your focal plane. In confocal microscope, you can also you can see the subcellular or cellular details with clarity. So that's kind of unique about confocal microscope. And because of this region, confocal microscopy and its advanced forms like two-photon microscope or second harmonic generation microscope, they're all being used as a fundamental research tool for biomedical, or chemistry researches in many areas. But we can also use confocal microscope for medicine. And what people have tried to do was take a look at the human skin with confocal microscope. So what you see is human skin imaged with confocal microscope, and these are the individual basal cells of the skin, and you can see the structures that are composed of these basal cells. So what's really nice about this image is that the image was taken in vivo, which means that we didn't have to remove the tissue from the skin and image it under microscope, but you can actually take a look at it while the tissue is still there, intact on your skin. So that's in vivo part. Other thing is among the many in vivo imaging technologies such as MRI, CT, X-ray, and all sorts of other things, confocal microscopy is one of the few which can actually visualize cellular details that might be associated with disease. And oftentimes, being able to visualize cellular details is really important in terms of cancer diagnosis or other disease diagnosis. So we have covered confocal microscope, which can do in vivo cellular imaging for human skin or human tissue in general. So in conventional method of conducting the confocal microscope, as we have seen in the schematic diagram, you take a look at one point at a time. And in order to generate an image, what you have to do is you have to move your imaging beam over the tissue using beam scanning devices, which are typically galvo scanner or acoustic opto modulator and things like that. In our approach, we are taking a different sort of method where we have a diffraction grating, and we hit the diffraction grating with broadband light, so that the light gets instantaneously dispersed into multiple beams, and different color or different wavelengths go to the different location of the tissue, so that we can image one line on the tissue with one single shot of the light. So that's kind of like order of magnitude faster than conventional approach. So by doing so, this particular axis or coordinate of the tissue is encoded with wavelengths. That's why it's called spectrally encoded imaging. As I just mentioned, by using this spectrally encoded imaging method, we are changing the imaging paradigm from serial imaging to parallel imaging. And then we can increase the imaging speed by 10 to 100 times. And there are quite many things that we can do with that high imaging speed. Other thing, too, is that since we are not using any sort of active mechanical scanner to collect light from the line area of the tissue, we can easily miniaturize the microscopic optics and use it for endoscopic imaging or other clinical applications. Depending on the bandwidth, so we are typically using relatively small bandwidths compared to the central wavelengths. So for instance, if we are using something like 1300 nanometer, the bandwidth is like 100 nanometers. So there definitely is a resolution variation, but it's not as dramatic as like in 2 times in higher. Please.
It's faster because until the blue beam gets to the red end of the tissue,
right.
You don't have a complete image of the red end of the tissue. And so it seems like it takes every bit as long to scan as
so, right. So, so here what we are trying to do is not to collect the spectroscopic data from the tissue. So each location of the tissue is sampled by just one wavelength. So when you shine broadband light into diffraction grating, and then they go together as a parallel sort of process on the tissue, and they collect it back. And then we use high-speed spectrometer or high-speed wavelength swab source to collect the data. So the one point on the tissue doesn't necessarily have to be scanned by all these wavelengths, but it's just scanned by one wavelength, which is there by just putting the broadband light into it. Does that make sense? Or
well, I understand.
Yeah.
It sounds though that it's not fair to call it say 10 times faster because you're not collecting the same information.
True, yeah. Correct, yeah, correct, yeah. So that is absolutely correct. And so because of this, there are some limitations. So for instance, we are not collecting spectroscopic data from the tissue. So there are applications that we can utilize this method, and there are other applications that we cannot actually utilize this method, or hard to utilize this method. So, but so today we're going to mostly talk about the reflected light coming back from the tissue, where the spectroscopic information is not that often used for clinical application. And in that scenario, spreading out this spectrum actually helps in terms of increasing the speed. So we have been doing 2 different major research areas using this approach. And the first one is something called comprehensive confocal endomicroscopy. So finding in a small size in the early stage in the tumor out of this gigantic organ is very similar to finding this Waldo guy from this BG market picture. So basically you have this BG picture, small pattern, and you're supposed to find it, right? So did anyone find Waldo by now, or here? No, here no. Okay, so I'm sure there are a couple of people who already found it. But so for some people this is easy, some people it's not. but eventually you're going to find it, right? So I mean, if you are given like 1 day to find it, you're going to find it, right? But in reality, though, I mean, for medical imaging, oftentimes there are imaging modalities such as MRI or CT where you can image the whole organ, but your resolution is not really there to visualize the details. Okay, so that's one end of the spectrum. The other end of the spectrum is where you can take a small piece of tissue out from the patient and then prepare it as a slide and then take a look at it under a microscope. Okay, but you can only do that for a very small fraction of the tissue, so you don't really have a big picture. So you have details, but you don't have big picture, right? So we have two different situations here. So, and that actually is the case. This is an example video endoscopic image of esophagus where you can see the difference between normal esophagus versus Barrett's esophagus. Pretty clearly, there's a color difference. And Barrett's esophagus is a pre-malignant condition to esophageal cancer. Okay, so it's easy to tell which part of the tissue is diseased and which part is normal. But then among the Barrett's esophagus tissue over here, it's hard to figure out which point has most malignant changes or even in small size cancer. It's hard to find it from the video endoscopy imaging. So what clinicians do as a standard of care, for now, is to take biopsies from 4 points every centimeters. And the size of this biopsy tissue is a few millimeters, very small. So in reality, what's happening is that less than 1% of the tissue that actually might have a risk of having malignant change or even esophageal cancer is examined under microscope. Okay. So we talked about confocal microscope, and there is a company that made a commercial confocal endomicroscope where you can take a look at GI organs using confocal microscope. And this is what it looks like. These are individual cells, which doesn't really, came out nicely. But bottom line is you can visualize, cellular features. But the problem still is that the field of view or image size is pretty small. It's less than millimeters. In this particular case, it's about 200 microns. And people do mosaic and stitching and other stuff, but even then, the image size is still smaller than a few millimeters. So we have this situation where the organ that might have a risk of having esophageal cancer or malignant change is this big, and the standard of care biopsy or confocal endomicroscope can only cover a very small fraction of the organ. So what we're trying to do with our technology is a comprehensive confocal endomicroscope to image the entire organ that might be under risk of having malignant change, and then generate data like that. And with that, what we can do is we can do Google Earth of esophagus, basically. So you have an entire distal esophagus, and then you zoom in until you actually see, start to see the individual cell nuclei. And how do we do that? So we talked about using grating to speed up. Right, so it's the same method where we have a fiber for delivering light in and out from the patient, and then we have a grating that does the line scanning, and then what comes back goes back to the fiber. And since we are not using any sort of beam scanning device, and we are using high-speed wavelength strap source, we can increase the imaging speed 10 to 100 times. In fact, we are 20 times faster than conventional confocal microscope. At least the one that we developed for endoscopy imaging thing. And another kind of key development area was to develop endoscopic probe with a small size. So here we have a fiber, and then the beam gets collimated, and then we figured out how to make the grating at small size, about the 2 millimeter size. And then there's objective lens, which we used aspheric singlet as a objective lens. And then what's coming back from the tissue goes back to the fiber and then goes out the patient and then detected by the system. Here, the size of this device is about 6 millimeters. And one thing that you might notice here is that the image size or the field of view that we can take a look at per one shot of the imaging is very small still, 280 micron in this case. But then since our imaging speed is so fast, what we can do is we can rotate the device and pull back to generate this screw helical kind of pattern to image very large area of the esophagus. So this is what it looks like in SolidWorks. Everything looks beautiful in SolidWorks. And then, inside of this tube, it rotates and, comes back. So what we did was we first placed something called guide wire that guides the introduction of the device into the esophagus. And then once it's positioned properly, it gets pulled back and then rotated to generate this helical scan. And then we do, large area imaging. Okay. So this is kind of data set that we're grabbing. The image size that you're looking at is about 7.5 centimeters, about this long of the esophagus. And then the width is 2.2 centimeters with 6.6 gigapixels, which is the largest confocal in vivo data ever recorded. And then the imaging time was just 3 minutes. So within 3 minutes, you can have a comprehensive scan of your esophagus or distal esophagus at cellular resolution. The way to look at this in the data is that the esophagus is a tube, and let's say you cut on the side and then unwrap it, and that's what you're looking at here. Okay. So we can talk a little bit more about that. This doesn't have autofocus, and we kind of carefully chose the tolerance between the rotating piece and the stationary piece in such a way that it has a dithering of a focal point, which is plus minus 50 micron. So which is still position the focal plane inside of the epithelium where we want to look at. But there was a challenge with that, and yeah, so it's a little bit harder to implement, but and also that's something that we have to work on later. But for now it doesn't. So when you zoom into this in a particular area, for instance, you can see this Barrett's esophagus sneaking in between the normal esophagus in the tissue, and then if you zoom in even more, what you see is that you get to see this Christmas tree, light bulb kind of thing going on here, which are normal cell nuclei. And on the right side, you start to see these dark cells on the surface of the epithelium, which are called goblet cells. So they're pretty similar to what you are actually seeing from the histopathologic slide that doctors use for diagnosis. So this is normal slide, and then on the right side you're seeing these goblet cells, and then being able to visualize this goblet cell is really important for diagnosing Barrett's esophagus. Sure.
Where you're withdrawn in this direction.
Right.
You're scanning
sideways. Yep.
Red to blue. Over what range is the red to blue dispersion on this kind of scale?
Right. So what you're seeing as one strip here is in one field of view of given by objective lens. Okay. So that's about 280 microns. So it's not like we are covering the whole 7.5 centimeter with one dispersion. So yeah.
So the top of that strip might be imaged with red, and the bottom of the strip blue.
Blue. So, but so red and blue are just purely a conceptual sort of visualization purpose. The actual wavelength used was 1300 nanometer, centered at 1300 nanometer, and then had plus minus 50 nanometer bandwidth. So it was all near infrared light. Oh, sorry, I was going to miss that. Yeah, sure.
So that is a field of view this way, 280.
Yep, 280 micron. Yeah.
Okay.
Mm-hmm.
What is a field of view this way?
That way. So that's based on the rotation of the device, which is 22 millimeters.
Okay, so
Yeah.
one shot
Mm-hmm.
will give you only one line, and then you reconstruct.
Right. So basically, the device is rotated like this way, so that the width of the image that it can cover by one rotation is about 22 millimeters. So
it's conventional confocal in that dimension, and the other one you've got these 280 micron strips.
right. So right there, yeah. All right. Another kind of sort of interesting development for this esophageal imaging was that my colleague Nima and I developed this capsule. So instead of using the endoscopic regular endoscopy device to be able to get into the esophagus, now we made this pier-sized and pier-shaped capsule. Basically, everything is same as the optics that I presented in a couple of slides ago, but it's just made as a kind of pier-shaped. And I'm going to tell you what why we did that. So basically, it's a size of 7 millimeters and 30 millimeters. It's kind of sort of big size omega-3, pill or something like that. And what you can do is you can ask the patient to swallow it. And once it's in, I know. I mean, yeah, people do that all the time. So, and then once it's in, because your esophagus thinks that this is just, something to eat, and it kind of pulls it down. So, and because of the peristaltic movement, the capsule device goes down, and then the operator, pulls it back to scan back, and then we do this, down and up, scanning 4 times to, cover a large area of esophagus. Okay. And I mean, see how happy he is. I mean, and there was $100 compensation, obviously. So, but anyway, so this is kind of image that we can get from this capsule device. So you can cover from esophagus to stomach, which is about 17 centimeter long, length. And then again, the esophagus is a tube, and then you cut on the side and then open, and that's how you look at this. Okay. For the stomach side, you get to see this body funding in mucosa kind of structure. And then, a little bit, close to esophagus, you get to see this in the gastric cardia, glandular structure. And for normal esophagus, without, contrast agent, you don't really see much. But then when you go into this island with higher, brightness, you get to see these small cells, which we validated through an ex vivo study that these are inflammatory cells, which are associated with this particular disease called EOE. So these are the kind of sort of things that we can do with this comprehensive.
When you spin this thing and you withdraw it, what you're actually recording is the spectrum as you
yes,
rotate.
Right.
And then afterwards you're reconstructing
right.
from that spectrum.
Right. That's correct. Yeah. So it's kind of sort of flip approach compared to conventional spectroscopy where you have a spectroscopy outside, right? But now we have a spectroscopy inside, and then spreading out, but also outside we have a spectroscopy to analyze it.
There's no need. So when you record that spectrum, which represents the 280 microns,
right?
Is there any reason you need to channelize that, or do you just keep that as an analog picture?
It's just an analog picture for now. So yeah. All right. So going back to the finding Waldo challenge. So now we have a technology where we can visualize, this whole picture with, all the details. Still, the challenge might be that, it's not all that straightforward to, find this in a Waldo guy or any sort of, pattern that we are interested in. So one thing that we can potentially do moving on is to highlight, some of the, features so that you can easily, identify this guy over here. Okay. So this is, what I used to do back in Massachusetts, and I will probably, continue working on this in other, with other, advanced forms of technology. So that's first part. And we switched the gear a little bit. We're going to talk about low-cost smartphone-based confocal microscope, which is I'm really excited about. So we're going to first start with the clinical challenges that folks have in Africa or some other low-resource settings. So here in the U.S. or any developed countries, the disease diagnosis is typically based on biopsy and microscopic exam of the tissue. So those two are really important, for any standard disease diagnosis in the U.S. In low resource setting, though, it's quite different story. So, for example, there's this disease called Kaposi sarcoma or KS, and it's one of the most common cancers in sub-Saharan African countries like Uganda or Kenya, things like that. So, patient comes to the local clinic with this skin lesion, and then the doctor, typically public health worker in the local clinic, doesn't have too much of diagnostic tools. So they have hands and eyes, and that's it. So they take a look at it, touch it, and then make diagnosis without any biopsy. Okay, and that often leads to a pretty high misdiagnosis, which is 33%. Based on one of the studies, and then that means that many of these patients are inadequately treated, and sometimes the patient dies because of the treatment, not because of the disease. So that's a huge problem there. And my friend and collaborator Esther Freeman at Massachusetts General Hospital And her team started this biopsy service in Uganda or Kenya and other African countries. And in this scenario, what happens is that the patient comes to the clinic, and then we take biopsy, and then biopsy sample goes to the central hospital, and it gets processed as a histologic slide. The doctor takes a look at it, and then generates the pathology report and comes back to the patient. And this whole process takes about 4 to 6 weeks. Okay. The challenge, though, is that by the time that diagnosis is made and delivered to the patient is not there anymore. Even for the local clinic, these folks have to walk hours and days to come to the local clinic, and it's not always straightforward to find that patient again and then start the treatment. So there's a huge delay and even a failure of timely treatment for this particular disease. So we since we are talking about confocal microscope, there are commercial confocal microscopy devices that have been used for human skin imaging. So for instance, what we can do with confocal microscope in terms of human skin imaging context is that you can actually see the difference between normal cell versus normal tissue versus melanoma, and it's pretty obvious. I mean, it's pretty in order where melanoma, in case of melanoma, it's pretty chaotic, and you lose some of the key features that normal tissues have. So and there have been several studies that validated accuracy of confocal microscope for many of the skin diseases. And it's currently being adopted in the U.S. clinic and other countries. So why don't we try to use confocal microscope in Uganda, let's say? Okay, so what's wrong with that, right? The problem is the cost of this device is about $50,000 to start with. Okay, so it's relatively expensive, and because of that reason, even in the U.S., I mean, very few, especially in the hospitals, have owned this device and actually utilize it for clinical practice. And why is it expensive? I mean, a couple of reasons. One is that it's using laser, which can be anywhere between $100 to maybe $1,000. And there's a beam scanning device, and it's associated in the controllers and circuits and things like that. And objective lens is another area where things can be pretty expensive. And then what comes back to the detector needs to be detected through a high bandwidth detector and DAQ system and computer and things like that. So all these things add up, okay? And then company has to make profit, obviously. So the cost is about $50,000 for now. And I think that's kind of low end. I mean, sometimes it's $100,000 depending on the version. So we talked about this grating trick, right? So we talked about how we can sort of spread light into multiple beams and then take a look at one line of the tissue without having a beam scanner. So that's kind of cool. That might help. But still the problem was that, I mean, especially with the endoscopy approach, that the device needs to be mechanically moved to generate a two-dimensional image, right? So that's one problem. The other problem was that I didn't mention, which is kind of my fault, but the kind of laser that we are using for this spectrally encoded confocal microscope is very specialized one. It has 100 kilohertz repetition rate and external cavity, polygon mirror, all sorts of things. So that's pretty expensive. And then the detector also needs to have a very high bandwidth with high sensitivity. So all these things add up so that the device that we have been using for endoscopic imaging is also somewhere between $50,000 to $100,000 range. So it's not really utilizable. So we came up with this idea where instead of using the pinhole aperture, which is commonly used for confocal imaging, why don't we use slit aperture? So let's say we have this in a very thin line where the light can sneak through. Then instead of illuminating one line on the sample, we can actually illuminate two-dimensional area, and that two-dimensional area is covered by multiple lines, and then each line has a particular color or wavelength. Okay, and then what's going back or reflected back from the tissue goes back to this detection slit where we can do confocal trick. And then after that, we kind of spread light again to generate this confocal image, and we just have to have two-dimensional imaging sensor right at this focal plane to collect the data. Okay, so we don't really need anything like beam scanning device, high bandwidth detector or DAQ and things like that. And this imaging sensor actually can be any imaging sensor within a high enough sensitivity, which means that this can be a smartphone, imaging sensor, and we can think about, making smartphone confocal microscope, which is. That's true, yes. So we compromise in actual resolution of the confocal imaging, which we can talk about later, I think. Yeah. So we actually made it. So basically, we used LED as a light source instead of laser, and this LED was like $8 or something. And then basically, it's similar to the schematic that we just saw before. The light is collimated and diffracted, and shine the tissue, comes back, and then filtered by the detection slit over here, and then respread, and then goes to this CMOS sensor, and then gets detected. Okay, and this is a photo of the actual device. And what's kind of interesting is that in terms of optics assembly, we have come up with this custom optical holder design, and then 3D printed them. And in one example is something like this. This is holding the collimation lens and prism and grating and the objective lens and things like that. And this is all in the 3D printed with right tolerance. I mean, I went through 20 different versions of this design to just make it work. And so now it's at the point that it's almost like an IKEA furniture where you just print this and then order things from Thorlabs and then pop things in, and that's it. So thanks to that, the material cost for this device was about $4,000. Okay, and one thing that I want to mention, though, is that the objective lens that I used for this first prototype was just over $2,000. So once we come up with a better objective lens design with low cost, I think we can also decrease the device cost quite a bit. And then obviously we have to test the resolution. So this pattern over here is about the width of 1 micron resolution. We measure the fluid half-max of the line spread function, which is about 2 micron. And then actual resolution was about 5 micron. And then considering that the cell size for skin imaging is somewhere between 10 to 20 microns, so this is probably sufficient resolution for imaging cells in the tissue. So we did in the human skin in vivo imaging, and what you're seeing on this particular image is spectral refraction from the tissue surface or skin surface from stratum corneum. If you go just under 40 to 50 micron, you start to see these dark cells, which are called keratinocytes. And in case of keratinocytes, the cell nuclei are not bright, but then cell membranes are bright. So you get to see the outline of the cell rather than the core of the cell. And in this case, if you go a little bit deeper, the cell size gets smaller, and then you start to see these bright cells, which are basal cells. And then once you go even deeper, you start to see these circular patterns, which is called dermal papilla, where the small capillary blood vessels comes up and down. Okay, so so basically the message is that this smartphone confocal microscope device can visualize cellular features in vivo, and that's first time ever with a smartphone device. And with that, what you can do is you can sort of do kind of navigate around different areas of the tissue, so that you take a look at one location, go up and down, and then so for instance these are capillary vessels right there, and then melanin deposition, and then if you come up a little bit, you get to see keratinocytes. You go other location, melanin deposition again, and things like that. Okay. In case of this particular video, though, the patient or the subject didn't have a skin disease, so you don't really see disease structure. And just in a recap of what we just saw, keratinocytes and melanin cap and capillaries that are visualized in the confocal image. So now that we have this device that can actually visualize cells in vivo from human skin, we can think about this new approach for diagnosing chaos. So in this new approach, the patient comes to the local clinic, and we use our device to take a look at the confocal image of the skin lesion. And that confocal image can be wirely transferred to an expert in the confocal reader anywhere in the world. And currently, in the study that we are doing in Uganda, the confocal image readers are in the US, one in Boston, one in New York, and then the third person is in Spain. So, the person can be anywhere, okay? And then they take a look at it and then make diagnosis, and the diagnosis can be communicated to the patient, by email or other, wireless method. So that's one way to go. But once we have enough of confocal images, and once we validate that this has a high enough of accuracy, then we can also think about automated image analysis method where we can develop a smartphone app and do the confocal diagnosis right at the spot, and then start the treatment. So either case, we can make the diagnosis right at the spot while the patient is still at the local clinic, and then we can initiate the proper adequate treatment right at the spot in timely manner. So in order to test this, I flew out to Uganda, and this is the hospital that I'm working with there, and this is my collaborator Esther and Agre, and we have delivered 2 smartphone confocal devices. As I mentioned, it's so relatively easy to make one of these devices. It takes about 1 week to make one. So we deliver 2, and then third one is in my office upstairs. And then after 24 hours of flight and several security check, I had to make sure that the resolution is still there. And then you see that these small patterns in group 8 and is pretty nicely visualized there. So with that device, what my clinical collaborators are doing is that they take a look at the skin lesion, and this person holding the confocal device and also doing the imaging is actually clinical technician rather than the doctor. Okay, so and then the kind of images that we can get is like this. So you get to see dermal papillae with melanin deposition around it, and this is a little bit blurry version of keratinocyte structure. But what's more interesting is coming out from the skin biopsy imaging ex vivo in Uganda, where we get to see these epidermal cells over here, and you can see collagen fiber structure or in adipose cells, and most importantly, we come across these kind of images quite often, which is that all these small holes are showing capillaries, and having irregular sized or shaped capillaries is a good indication that, or right indication that the patient might have a tumor, skin tumor. So we're still collecting the data, and then also the testing out the diagnostic accuracy with our device.
What wavelength range is this device?
This one is centered at 509 nanometer. It's amber-ish color with a bandwidth of 80 nanometer. Okay. So still, it's not quite red to blue, so it's a little bit misleading. That's true. So it goes about epidermis and a little bit of top of the dermis. So that's a little bit of challenge for some of the diseases. But many of the skin diseases actually arise from the epidermis area. So being able to take a look at the boundary between epidermis and dermis is important. And this might be able to do it for certain cases.
Why didn't you choose longer but still to be detected by the cell phone?
Right, true. So one of the reasons to use this particular wavelength was that, so as we understand the field of view size or the image size that we can cover with our approach is determined by the bandwidth of the source. And so the particular LED that I have used is 80 nanometer, and that's by far the largest among the commercially available low cost LED sources. If we go somewhere like 630 nanometer, the bandwidth might be 30 to 40 nanometer, which cuts down the field of view quite a bit. But we can definitely figure something out, because then you can increase the group density so that you spread out more, right? So kind of do the trick there. So, but you're right. So for now, just because of the 509 nanometer, there's some limitation because of due to the penetration. So we are continuing collecting data from Uganda and trying to figure out, what we can do in terms of diagnostic accuracy. But what else can you do? What else can you do with this device? So one thing that I'm eager to do is to develop an endoscopy device that works with a smartphone confocal, and then take a look at cervical cancer, which is the most common cancer in Uganda for female population. So I recently submitted NIH grant application for this, and keeping my fingers crossed. So hoping for the best. But not only that, so I briefly mentioned that even in the US, not many clinics actually use this, in vivo microscopy device. And one of the main reasons is the cost, okay? So now, if we can actually, make these, low-cost device available, then we can think about using it in, rural clinic or in a primary care clinic to, screen very large population and then catch the skin cancer at early stage and then treat them, accordingly. So that's, really a big opportunity right there. And last thing, which is very interesting is that, we can think about using this device as a medical education tool for pathology or dermatology, and also in STEM, education in K-12 or college education. So that's going to be all pretty interesting. So with that, I would like to thank my collaborators and mentors, all over the world, and funding agencies. And one thing really rewarding was to work with a team of really nice and talented, passionate engineers and scientists and doctors. Okay, so the teamwork is the key. So among the students, if you're interested, join the team. Okay, so we might not as cool as in Avengers, but we're going to have a lot of fun and make something that's useful. So I'm this guy actually. So taken. Okay, so all right. Thank you for your attention. Sure, yeah.
The department head is standing behind you?
Oh, that can be you. Oh, yeah, Tom, Tom could be.